Today's lesson is going to go over the defining neuropathology and anatomy for the neurodegenerative diseases that we discussed in the first class. There's a lot to cover, so this video is going to be a bit longer, but I promise it'll be the longest one in this section. As a reminder, these are the four subdivisions that I brought up before: tauopathies, synucleinopathies, trinucleotide repeats, and prion diseases. Before I mentioned that each of these families and that Before, I mentioned that each of these families and their names derive from the proteins that characterize them: tau, synuclein, and prion. Trinucleotide repeats are somewhat unique in that this refers to the DNA rather than the protein. I want to show this table again just to remind everyone how different neurodegenerative diseases are characterized. I do highly encourage you to take a look at some of these other diseases that I don't have time to cover, all of these ones down here, since I will only be discussing the first three that we introduced in the last lecture. So let's jump right in with Alzheimer's disease and take a look at the pathology and what is known as Braak staging. Alzheimer's, as we know, is characterized as a tauopathy, which of course means that tau is prominent in this disease. You may have heard the term neurofibrillary tangles or tangles in reference to Alzheimer's disease. And this is in reference to tau, which this black arrow is pointing at over here. There is another key aspect to Alzheimer's disease, though, that you may have heard of as plaques. Alzheimer's is not only a tauopathy. It can also be defined and characterized by the presence of these neuritic plaques, which are made of amyloid beta. The white arrow here is pointing to an amyloid beta plaque. Now, I mentioned in the last class that an official diagnosis is not made until after a patient dies. In order for a person to be confirmed to have had Alzheimer's disease during life, a pathologist must see both tau tangles and amyloid beta plaques. Importantly, the tau accumulations are intracellular, meaning they are inside the cell, which you can kind of see here with the cell body and a little bit of an axon protruding. The amyloid beta plaques, in contrast, are extracellular, meaning they accumulate outside or between the cells. And before we jump into some specifics about where we see these accumulations in the brain, let's take a look at what exactly these proteins are. Tau is great because it is very clear and well known what it does. In a healthy neuron, tau is important for binding microtubules and maintaining the structure of the cell, specifically in axons. You can imagine then how tau turning dysfunctional can be devastating to a cell. So in a healthy, normal axon, we see tau maintaining this microtubule structure. However, loss of functional tau leads to a diseased neuron where microtubules start to dissociate and tau starts to become sticky, sequestering in itself and filling up the cell, leading to those intracellular tangles that we see, these neurofibrillary tangles. Taking a look at amyloid beta, it is not quite as straightforward as tau. In order to understand amyloid beta, we need to back up to another protein called APP. And there's a lot going on here, but we're going to walk through this together. APP here is the amyloid precursor protein. And in healthy cells, APP does quite a lot. It's really important early in development, aiding in synaptogenesis, or the birth of new synapses, neuronal growth, and cell adhesion. Again, I'm sure you can see why dysfunctional APP would be bad, looking at the list of important functions it has. Because interrupting the creation of new synapses and disrupting cell adhesion would lead to an interruption in normal communication between cells. APP is not the defining protein of Alzheimer's disease, though. It's in the name that this is a precursor protein, which is where APP cleavage becomes very important. So APP can get cleaved or cut in a couple different ways. On this left side over here, we see that alpha secretase can come in and cut APP here, leading to two different components, one maintaining this transmembrane location. Gamma secretase will then come in and cut right here, right in the center, leading to these other fragments. This does not lead to amyloid. On the other side, though, we have beta secretase that can cut APP up here, leading to these three components being left over, where gamma secretase comes in again and cuts in the same spot, leading to these two components, amyloid beta and this other component that we don't really care about here. Amyloid beta then aggregates, and the tricky part is that we need amyloid beta. We need to have this in order to lead to these physiologically good functions of amyloid beta, leading to synaptic plasticity, neurogenesis, and even working as an antioxidant. The problem comes in when the body starts to make too much amyloid beta. And instead of following this healthy pathway up here, we start to lead to aggregation of amyloid beta in a negative way, leading to the formation of what we call fibrils, or long stretches of sticky amyloid beta, which will eventually become plaques and leading to neuronal dysfunction and cell death. People will often talk about fragment sizes of amyloid beta, so you may hear reference to amyloid beta 42 and amyloid beta 40. It's thought that amyloid beta 42 specifically seems to be this stickier variant of amyloid beta, leading to more toxic accumulation. So sometimes levels of amyloid beta 42 are used as a biomarker to suggest Alzheimer's disease. And although Alzheimer's disease is classified as a tauopathy, most of the mutations we know that lead to the genetic causes of Alzheimer's are on this APP gene. This graphic is a little bit older. If you notice, it's from 2003. But many APP mutations have been identified. You can see several of them listed down here. And they are named based on the location that the families come from. So Flemish, Iowa, London. Most of these genetic causes of Alzheimer's disease are classified as early onset, whereas sporadic diagnoses tend to occur at older ages. So if a person has one of these mutations, they will likely develop the disease before the age of 65. One thing that is certain is that they will develop the disease at some point. There are some other genes that are known to lead to Alzheimer's disease, which I'm happy to talk about during our synchronous class time if people are interested. Zooming out a bit, I want you to recall the definition of neurodegeneration that I gave you as CNS cells losing function over time. These proteins accumulate, which means that it doesn't happen all at once. This schematic here shows a little bit of the macro view of the progression we see from healthy all the way to severe Alzheimer's. And I want you to take note of the initial pathology that pops up in preclinical Alzheimer's and MCI, or mild cognitive impairment. You can see that this really centers on the temporal cortex here, which then spreads into pretty far regions of the brain once we get into mild and moderate Alzheimer's disease, and severe Alzheimer's disease is full, pretty much neuropathology everywhere. There was a point in time when this progressive nature of pathology was not known, though. So I want to introduce Drs. Heiko and Eva Brock, a dynamic duo in science. They were the first to propose this staged progression, specifically with neurofibrillary tangles, back in 1991. This is Dr. Heiko and this is Dr. Eva. I could not find a photo of Dr. Eva online, but there are many, many photos of Dr. Heiko. Because they are the first ones to make this splash, we now refer to the stages as Brock stages. So let's jump back into the brain regions. And I want to show you the first three stages of the Brock stages related to Alzheimer's disease. The dark areas down here are the areas indicating tau accumulation. As I just mentioned, we see these early stages where tau starts to accumulate a lot in the temporal cortex. And specifically, we start to see the rhinal cortex spreading into the hippocampus and other temporal association regions early in this disease. Let's think back a little bit about the symptoms of Alzheimer's disease. Remember, it's highly characterized by memory loss, confusion, communication struggles, changes in mood, changes in vision. And it starts to make a lot of sense when we look at the function of these brain regions. So the rhinal cortex is essential for unifying and passing along information. The rhinal cortex can be broken down into a few subregions, like the perirhinal cortex and the entorhinal cortex. And you can see here its positioning between the hippocampus and the cortical areas. So proper functioning of the rhinal cortex is essential for hippocampal to cortical functioning in both directions. The hippocampus is a region that you should be pretty familiar with by now as being important for spatial navigation. It is also critical for memory formation, especially working memory. And finally, the temporal cortex is essential for integrating and storing memory. Moving into the further stages of Alzheimer's disease, looking at stages 4 and 5, we start to see spread of tau into the neocortex, where we see storage of long-term memory, which lends to a sense of identity, as well as the location of many of our senses, such as vision. Diagnosis of Alzheimer's typically occurs during stages 3 to 4, and this is after years of tau accumulation in these early brain regions. By the time we get into stages 6 and 7, pathology is seen pretty much everywhere. Now, those were all images depicting tau accumulation, but amyloid beta follows a very similar progression and timeline. Across the top here, we see the progression of tau in a shortened schematic, and along the bottom are the amyloid beta accumulation. We can see that it starts in the same regions as tau and spreads in a very similar way. And I again want to highlight that diagnosis typically occurs during stages 3 to 4, and point out that amyloid beta pathology is already fairly widespread by the time a person would actually be diagnosed in the clinic. Let's move on to the pathology and progression of Parkinson's disease, the synucleinopathy that we are focusing on. Parkinson's disease is characterized by the presence of what we call Lewy bodies and Lewy neurites, which you can see in these images here. This top image is a Lewy body. So this pink circle inside of a darkly pigmented dopamine cell. And this pink squiggle down here is a Lewy neurite. These are primarily made of a protein called alpha-synuclein, specifically alpha-synuclein that has been phosphorylated. Alpha-synuclein is a relatively small protein, which is depicted in this graphic. Interestingly, if you pay attention to the small text here, alpha-synuclein also plays a role in Alzheimer's disease, but it's secondary to the proteins that we just previously talked about. In Parkinson's disease, we see that phosphorylation here at serine 129 leads to alpha-synuclein becoming very sticky, accumulating on itself, and forming these dense intracellular aggregates. This protein was identified to be of interest in Parkinson's because of the mutations in this gene that led to familial Parkinson's disease, which we can see a couple listed here. Like with APP, there are several mutations from different families that have been identified. And since this graphic came out in 2008, there have actually been two more mutations identified that are also over here on this alpha-synuclein gene called SNCA. Like with Alzheimer's disease, familial cases are typically considered to lead to early onset Parkinson's disease compared to sporadic cases, which typically have a later age of diagnosis. Looking at where in the brain we see pathology, there is a similar progression over time, and I bet you can guess who described this staging. The location and progression of Lewy bodies looks pretty distinct from tau and amyloid beta, though. In early preclinical stages of Parkinson's, Lewy bodies are seen deep in the brainstem and in the olfactory bulb. Entering into stages 3 and 4, which is when patients are typically diagnosed, Lewy bodies are seen in lower regions down here near the hypothalamus and thalamus in the midbrain. And by late stages of the disease, stages 5 and 6, Lewy bodies will have progressed into areas of the cortex. I want to get a little more specific though, and look at the brain regions that are most affected during these stages when diagnosis occurs. This is a cross-section of a human brain. Down here is the brainstem. Here is the striatum, and up here is the motor cortex. In preclinical Parkinson's disease, we start to see Lewy bodies deep in the brainstem, working their way up into the midbrain and interbrain. One interesting fact about Lewy bodies is that they love dopamine cells. We are not totally sure why, but they work their way up into this region here called the substantia nigra. The substantia nigra is full of dopamine cells, and these dopamine cells have a dark pigment, which naturally makes this brain region darker. The substantia nigra is small, but it's highly impactful. It communicates and regulates back and forth with the striatum, which in turn communicates with the motor cortex to regulate all of our movement. Together, this circuit is known as the basal ganglia, which you may have heard of. When Lewy bodies take hold in the substantia nigra, this throws off the regulation and communication with the striatum, which in turn throws the entire motor system out of whack. One interesting aspect that I want to highlight is we tend to see Lewy bodies take hold in one hemisphere sooner than the other, which brings me back to the symptoms we learned about in the last class. So remember, Parkinson's symptoms include bradykinesia, a unilateral tremor, facial motor difficulties like apathy, gait changes, and sleep disturbances. And we see pathology primarily in the brainstem, substantia nigra, and striatum when patients come into the clinic. The brainstem, of course, is important for autonomic function in controlling our craniofacial nerves. As I just mentioned, the substantia nigra is important in controlling striatal signaling, and the striatum, in turn, controls motor output, and importantly, motor suppression, or telling your muscles to not move when you are trying to sit still and listen to a lecture or watch a video. This graphic just depicts what occurs in Parkinson's disease, where we see a loss of dopamine neurons, leading to an interruption of the normal inhibitory regulation in the striatum. Currently, treatments aim at targeting this location, but we'll talk about treatments more when we get to our class on treatments and future directions. I mentioned in the last class that I talk a bit about pesticides and toxic chemicals here. You may notice that this box says Parkinsonism. This term refers to both Parkinson's disease itself and disorders that look like Parkinson's disease or Parkinsonian-like diseases. Exposure to pesticides like paraquat or rotenone, or to toxic chemicals like a chemical called MPTP, lead to the rapid degeneration of dopamine cells in the substantia nigra. So people who encounter these substances will present with Parkinsonism or Parkinson's-like disease. But I want to highlight that these are distinct from actual Parkinson's disease as a neurodegenerative disease because they do not show accumulation of Lewy bodies that are necessary for a diagnosis of actual Parkinson's. The symptoms look the same, but the underlying pathology is different. Again, I want to bring us back to this working definition we have, that CNS cells lose function over time. A big question, though, has been how does this over time piece occur? How does this pathology spread? And we still don't have all the answers to this question, but I want to talk about one hypothesis that has been gaining evidence for several years now. And this is the prion-like hypothesis, which was first described by this man here, Dr. Stanley Prusiner. Prion stands for proteinaceous infectious entities. We aren't able to talk much about strict prion diseases in this class, but prion diseases have known infection points and cell-to-cell transmission via the prion receptors that lead to toxic aggregation. Dr. Prusiner studied a lot of prion diseases and suggested that other neurodegenerative diseases may work in a similar or prion-like fashion where misfolded proteins spread from cell to cell. This idea then is that at some point, a person may come into contact with alpha-synuclein that is misfolded or in a sticky, fibro-like state. This sticky alpha-synuclein enters a body that is full of natural, healthy alpha-synuclein, which then starts to stick to it. In turn, the native alpha-synuclein starts to phosphorylate, becoming sticky itself and attaching to other phosphorylated synucleins. It is thought that the immune system at some point is able to eventually get rid of the original fibrillized alpha-synuclein, but not until it's too late and a bunch of the other natural alpha-synucleins have jumped onto the phosphorylation bandwagon, which then leads to full-blown Lewy body accumulation. I've used the example of alpha-synuclein here, but the same idea is thought to apply to tau and amyloid beta as well. Some evidence that has led us to think that this may be the case comes from the earliest Braak stages in Parkinson's disease. Recall that we see early accumulation in the olfactory bulbs, as well as deep in the brainstem, which has led research to explore if it's possible that people being exposed to something via the olfactory system or ingesting something where the enteric nervous system or the nervous system of the gut takes up the fibrillized protein may be the way in which this external misfolded protein is introduced to the body, which then leads to accumulation spreading up the brainstem or spreading through the olfactory bulbs, eventually leading to the slow accumulation of Lewy bodies in Parkinson's disease. So far, experiments in animals are showing that this is pretty likely, which has totally changed how we are thinking about these diseases and how we could possibly treat them. To wrap up today's lesson, we'll take a quick look at Huntington's disease, our trinucleotide repeat example. As I mentioned in the first lecture, Huntington's is a disorder with a known genetic cause, this CAG expansion on the Huntington gene, which leads to translation into a misfolded protein that aggregates inside cells. You can see this in these top two panels here, with the green and the red showing Huntington aggregates inside the blue cells, compared to a control age-matched person where we don't see any green or red accumulations. These accumulations dramatically affect a circuit that we touched on earlier, the basal ganglia. Now, I'm going to expand a bit on that circuit, and I don't need you to know all of the fine nitty-gritty details of this, but I do want to walk you through it. Here, I'm showing you a schematic that highlights more in depth the regions that make up this circuit. In a normal, healthy, functioning individual, there is communication occurring from top to bottom, cortex down, as well as from bottom up, from lower brain regions back up to the cortex. The cortex, especially the motor cortex, sends signals into the striatum, made up of the caudate and putamen. The substantia nigra also sends signals into the striatum. These two signals, the cortex and the substantia nigra, affect the output of the striatum, going into the globus pallidus external or the globus pallidus internal. In turn, these have downstream effects on the thalamus, which communicates back up into the motor cortex to either promote or inhibit movement. The two pathways that we see here in the basal ganglia are known as the indirect pathway, shown on this side of the dotted line, and the direct pathway, shown on this side. The indirect pathway inhibits movement, and I remember this because of the two I's, indirect, inhibit. The direct pathway, conversely, I'm sure you can guess, promotes movement. That is all a lot of information, and it's a very complicated circuit that I went through kind of quickly. But the important thing that I want you to remember is here, when things start to go haywire. On the left is a simplified version of what I just showed you, a functional, healthy basal ganglia. And we already discussed what Parkinsonism looks like, where we see severe degeneration of the substantia nigra, which eliminates important modulating signals into the striatum, shown by the dotted lines moving up here, which then activates the indirect pathway too much. You see this thickening of this black arrow here, leading to a hypokinetic disease, or less movement. On the flip side, we see diseases like Huntington's disease or chorea diseases. Huntington's, the direct pathway becomes overactivated because the striatum itself is starting to degenerate, leading to this hyperkinetic disease. You can remember this then as the direct pathway leads to dancing. To compare a bit of what we just learned, because we just learned a lot, here is a table that highlights the primary proteins, genes that when mutated are known to lead to these diseases, and brain regions that have pathology in early stages of diagnosis. In our next lesson, we'll review how we got to where we are in research and discuss different models that have led us to groundbreaking discoveries.